Hey friends, welcome to the 2024 AEOP Internships and Fellowships Speaker Series. We are so happy you are here to join us today. My name's Heather. I'm the Assistant Director of AEOP Internships and Fellowships, and it's my pleasure to host today's session and introduce you to an exciting series of talks that will bring our community together to explore opportunities and pathways of STEM. The AEOP Speaker Series is designed to foster community and learning. Through this series, we aim to introduce you to the opportunities available within STEM and provide insights into various career pathways. Our goal is to encourage you to take full advantage of AEOP internships and fellowships and everything that we have to offer. First, we'd like to just remind you that we have some master classes available in the AEOP Learning Hub. Everyone should have been invited and also attending those masterclass webinars. Remember, we also have that leadership opportunity, certificate opportunity, and any next step opportunities that you have for our speaker series will also be posted. So to kick things off, we want you to let us know where you're from. AEOP interns and fellows are conducting research all over the country, and let us know in the chat where you are from. We'd like to see. Like to hear and or like to see. Anybody want to let us know? Oh, there we go. I see some names now. Crystal, you got me with the Rochester. Go Bills. Great job. Thanks everyone for sharing. Keep them coming. Let us know if you have a group of people with you also. If you're logging in with multiple people, we would love to know how many people are with you. So we hope that this is just the beginning of the engagement today. As we are using the webinar function of Zoom, you will not be able to turn on your camera or voice. However, you can communicate informally through chat. And for any questions you have for any of our speakers, please use the Q&A function. That's where we'll be pulling questions to ask the presenters today. Before we dive into our session, we have a fun announcement. We will be selecting 4 random people during the session to receive an AEOP Pocket Scientist or Engineer. So these are fun, cool items that we'll post pictures in the chat that you can use while you're at your intern site or in the future, whatever you plan to do. So next, I would like to introduce Crystal. Crystal is an undergraduate communications intern with the RIT administrative team this summer. Crystal, it's all you.
Thanks, Heather. Good afternoon, everybody. Today, I will take some time at the top of every one of our speaker sessions to introduce opportunities available to you. Today, we'll hear from Andrea Milena, the JSHS project manager. She's based at the National Science Teaching Association, which is the administrating agency for JSHS since October 2021. Prior to joining NSTA, she worked in product management and finance roles with the International Training and Education Center for Health in Botswana and at the Mayo Clinic. A highlight of her career was the 4 years she spent as a Peace Corps volunteer in Botswana. Her favorite part of JSHS is interacting with the talented students. Take it away, Andrea.
Thanks so much, everyone. I appreciate the introduction. Like I said, my name is Andrea Milena. I'm the JSHS project manager for the Junior Science and Humanities Symposium, which is a high school program. Let me just get my screen shared and we will get started. Although it's starting out on the first page. But awesome. Okay. So Junior Science and Humanities Symposium, some of you may have already heard of this before, but we'll just call it JSHS for short. JSHS is a high school STEM research competition. It ties in very well with the various internship and fellowship programs going on, especially high school internships. You're very easily able to turn what you're doing this summer into a JSHS submission in the fall, and we encourage that. But what it is it's a regional, we have regional and national events where you're able to win university scholarships. It is an individual competition, and of course it is free, as is all of these lovely programs. And we're excited to be getting ready to celebrate our 63rd year. JSHS has a multitude of sponsors and administering agencies. We are sponsored in part by all 4 of the military branches, as you can see on the slide here. as well as our regional host organizations, which are typically universities throughout the US, and we do have 49 regions. So who can compete? Any 9th to 12th grade student is eligible to compete as long as you are a US citizen or legal permanent resident. Does not depend where you live or what type of school you are attending. We do have opportunities throughout all 50 states, US territories, the DODEA schools internationally. And even if someone happens to be living overseas, we also have opportunities for you as well. Some of the main benefits to the JSHS program, as I said, it is a scholarship competition. So we offer many scholarships. We pay out over $400,000 annually in scholarships at the regional level as well as our national level. We also give out cash prizes at our national level as well. And I'll go into more detail on these benefits on our upcoming slides. So just a bit of the timeline so you know what to expect when. You have expect students to be doing your research or developing your engineering design anytime. And then registration typically opens in October. And at that time, you will need to have your abstract and research paper ready. Regional events occur for competition in January and February, with our national event typically in April or early May. So within that research, something you can do at home during an internship, just as long as it's something that's been conducted and completed within the year of submission, you are allowed to submit research findings to multiple organizations. You know, if you're submitting to us, it's perfectly fine if you've also submitted to someone else. We do have a few other documents, such as our core rules and a statement of outside assistance that goes into a lot of detail about what's needed and the proper formats, and those are all available on our website. So I won't go into depth there. Just want to keep this very high level right now, but you can find all of those details at www.jshs.org. At our regional levels, you know, students compete together, but we do typically talk about the categories and how we break things out. At the regional level, students are competing just for the overall top first, second, third place. But once you get to nationals, we do break them out by these categories. And as it says at the top, you know, students who have found something of a social interest or that primary concern to themselves typically go quite far because of the personal impact. Registrations open in late September, early October. The links are always available on our website, again, jshs.org. We have a Find Your Region page where you would find what state your school is located. So the registration is dependent upon where your high school is located. And then you would complete the registration before the deadline. And then, yeah, then we have that opportunity if you get invited after the review process. We do have a virtual region as well, and this is intended for students who are not near their host organization's locale and cannot get to it, or if you know you're going to be moving during the school year since it is based on your school's location. Again, abstracts, papers are part of the submission and the SOA. We also have standardized judging rubrics, and again, that's also located on our website. So that's a good guide to help identify, you know, if you're getting everything that's needed into your submissions. We do posters and oral competitions. Those are highlighted again in core rules, and you can be invited to both or just one depending on where you're located. Regional events are January through February. And here's probably what a lot of you are wanting to know: how much money can I win? So at the regional level, every region of our 49 regions offers 3 prizes, $2,000, $1,500, and $1,000. Those are scholarships that would be paid directly to your university once you get to that stage. We also invite all 5 students, the top 5 students from each region, to our national event, and that is an all expense paid trip. So oral competition is, as it says, oral competition, slide deck presentation to a panel of judges, and poster competition. Again, details on the website. I'm just tracking time, so I'm going to go through these photos, but this will be a video you can watch later as well if you want to pause and look at some of these videos, pictures from some of our regional events this year. We have our Great Plains, which is Nebraska, Oklahoma, Kansas, Florida. We have our Puerto Rico region represented here as well. At our national event, as I said, all of our students, top 5 from each region, compete at the national level. And here it's really awesome. We now have it divided out into the 8 categories where students are, we could now win $12,000 in scholarships. And we give out 24 scholarships at the national level, as well as the opportunity to interact with a lot of amazing DoD STEM individuals, like-minded students, and have a lot of fun activities during the event. There's a few pictures of this last year that was held in Albuquerque, New Mexico. So JSHS is also great because we don't just, yes, it's the competition and that is our main focus. We also offer other opportunities and resources to our participants to help, make things as useful for you as possible. So we do have a virtual mentoring platform that is a free platform for JSHS participants to utilize. And we do vet the STEM professionals who are the mentors. And we also have research and presentation tips on our YouTube channel, as well as past abstracts. So again, go to our website. You can find so much there. And here is, all the ways to follow JSHS and learn as much as you can. It's a great opportunity, and we encourage everybody to participate. Yeah.
Thanks so much.
Yeah, I'll leave it on here. So if someone's wanting to track that for a minute. And thank you.
Thanks so much, Andrea and Milenia. As mentioned, if you're interested in any of the opportunities presented by JSHS, make sure to find your region on the website and follow their social media. If you have any questions, feel free to ask them in the chat. And I am now super excited to introduce our fantastic speaker today, Professor Cory O Hern from Yale University. He'll be presenting on answers on how to cure cancer and why go to graduate school in STEM, all in one talk, and his journey is truly inspiring. Professor Cory O Hern joined Yale in 2002 after postdoctoral fellowships at the University of Chicago and UCLA, and has become a leading expert in soft matter and biological sciences. He holds joint appointments in mechanical engineering and materials science, applied physics, and computational biology and bioinformatics. He's shown outstanding leadership, mentoring a research group of 15 PhD students and 4 postdocs. His work, supported by major institutions like the NSF, NIH, focuses on computational protein design, machine learning, and biomedical data and cancer modeling. Professor O'Hern has published over 150 peer-reviewed manuscripts and also more than 250 presentations worldwide. He's a fellow of the American Physical Society and a leader in the soft matter research community. Prepare to be inspired and gain valuable insights from Professor Cory O Hern. Take it away.
Thank you. Can everyone hear me?
Yep.
Okay, so first I want to give a shout out to my 9 high school students, my AEOP students that are watching me in New Haven, Connecticut. So good to see you're all attending. And what I want to do today is give you a story about my journey to where I am today, sort of convince you to go get a PhD in a STEM field. And hopefully I'll have time to talk to you a little bit about research. So the first thing is I'm a professor of mechanical engineering at Yale University, and then I have a bunch of other affiliations there. And that's sort of also the part of the story today, that to tackle complex problems of today, research questions of today, you need to bring a lot of different perspectives. So we need a physics perspective, we need an engineering perspective, and we need biology perspectives. And so I've taken on leadership positions to help in the training of those sort of merging perspectives at Yale University. Another aspect of this slide that was mentioned by Crystal is that it takes a lot of funding to do research to support the personnel. And so this work is funded by the National Science Foundation, the Army Research Office, and the NIH. The other thing that I want to do on the first slide is just really compliment and brag about the AEOP. So I've been involved with the AEOP for at least 5 years, and it's only been getting better. So the leadership team is doing a fantastic job. I think it's a great program for students and for getting students excited about science and research. Yeah, so the first thing, I want to give you a perspective of what it's like to be a scientist. And so one of the perspectives and one of the reasons why I went into science is because it's not a lone sport. It's not a lone occupation. It's done, and maybe you don't think about this as a scientist. Science is really a human endeavor. You have to explain concepts, difficult concepts to other people, because if you figure something out, but then you can't communicate it to anyone else, then it's useless. You want to make breakthroughs in science that can lead to future technological advances, medical advances, etc. So you have to be able to communicate your results and receive input from others to maybe improve your science. And so the other aspect here is that science is sort of intergenerational. So in the center, there are the old people, but then they're surrounded by a bunch of younger people. So my group is made up of around 30 people, and it spans an age from the PI, postdoctoral researchers, PhD students, undergraduate, and high school students. And so I enjoy the multiple perspectives from young to old. So in this talk, I'm also going to give you a picture into my life's journey into science. And so it started fairly early. I wasn't born in Florida, but we moved to Florida right when the Space Center. So everybody thinks that the Kennedy Space Center or the shuttle takes off from Cape Canaveral. That's just where they monitor they monitored the shuttle program. So the Kennedy Space Center is actually on Merritt Island, which is an island in Florida on the East Coast, a barrier island on the Atlantic coast. And so we moved there in the 70s when the space program was just taking off, and I graduated from Merritt Island High School in 1990. And kind of a funny thing is you can either have a good physics teacher or a bad physics teacher. I had a bad physics teacher, but despite that, I became a physicist and majored and got a PhD in physics. So as an academic, I'm going to talk about that it takes a lot of grit and time. And so this is my academic journey, and you get to see a lot of America. So I started, as I said before, I started in the East Coast of Florida, graduated from high school in 1990, and then started at Duke University, got a bachelor's degree in physics. I graduated in 1994 and then moved up to Philadelphia at the University of Pennsylvania. I stayed there for 5 years, got a PhD in physics, leaving in 1999. And then I did sort of a joint postdoc, and I'll tell you a little bit more about what that means. That's basically you need training after your PhD, sort of waiting for a job to open up as a professor. And so I did postdoctoral training at the University of Chicago and at University of California, Los Angeles. So my journey is sort of from Florida to the East Coast, then across to the West Coast, and then back to Yale University in 2002. And I've been at Yale since then for 22 years. So this is just sort of a chart. I just want to emphasize, and this is not necessarily a bad thing, but it takes, if you want to become a professor or a scientist, it does take a significant amount of training. So you have to graduate from high school, then you major in a STEM discipline. Typically that takes 4 years. Then if you want to go the PhD route, that takes typically 5 or 6 years. And then usually there's not a professorship that's available right after your PhD. You do some type of postdoctoral training, and mine was 3 years. And so that totals 12 years before I had a permanent position. And then after that 12 years, which was lots of fun, I've been at Yale for 22 years now. Time has flown by. So the type of research that I do is purely computational. So maybe that is somewhat new to most of you, that there's sort of three general types or two large categories of research. There's experimental, and that could be experiments on animal models like mice, if you're in the biomedical field, or it could be sort of in vitro systems, which are sort of model experimental systems, but it's wet lab work where you're using your hands and you're doing experiments. And then there's another domain, which is theoretical or computational, where a lot of your work is done on a computer. And so I experimented with both types of research, both experimental and computational, and learned that I was much more skilled at computational and theoretical research. One reason why I like computational work is that the topics that you can study are varied and wide. Whereas experiments, they can be a little bit more limiting because it takes so much money and time to build an experiment and develop expertise in a particular area. Whereas as long as you can fund or buy sufficient computational time or computational resources, then in principle, you can study any type of system that you want. And so these are snippets of the research that I do in my laboratory, or my computational laboratory. Protein design, studies of the development of leaf tissue, both flowers and plant tissue, both flowers and leaves, collective cell motion in spheroids, tumoroids, and epithelial tissue. In the bottom left, cancer invasion, which I'll get to today, and then flows sort of microfluidic, computational studies of microfluidic channels. So here's something that you may not have thought about, but what are the sort of rewards, or how do you know that you're being successful in an academic career? So sort of the currency in academics, isn't money necessarily, it's citations. And so you want your work to be highly cited so that you're communicating it effectively to other people. They find that your work is interesting and important, and then they will cite your papers. So we mentioned at the top that I've written 150 papers and counting. Some of those are more cited than others. So typically, a paper is sort of highly cited if it has 100 citations in STEM fields. My most highly cited paper has, I don't know, depending on how you think of it, only around 2,000 citations. Another thing to think about is sort of a record of how you're doing is an H-index. So an H-index is the number of papers that have that many citations. So as your H-index grows, it sort of becomes harder and harder to increase your H-index because you need more and more citations. So my H-index is currently 47. So I have 47 publications that at least have 47 citations. And, I can pat myself on the back, but, as you all know, there's always people who are doing really well. And so one of my mentors from University of Pennsylvania, who's now at Harvard University, sort of a leader in the field, is Dave Waits. And he has an H-index of 212 and 164,000 citations, whereas I only had around 10 or 11,000 citations, but something to aspire to. So one thing that I really like about the job is that, in order to get citations and get people to know about your work, you want to go and talk about it. You want to go and talk about your latest results, latest advances. And all of this is not funded by you personally. This is funded by grants that you bring in, or grants that you can get from your local university. And here's just a map of all the places I've been in the last, say, 15 years. So all over the East Coast, on the West Coast, And so I've gotten to travel across the US and, convince other scientists that my work is really good and that I have something important to say. And not only do you travel in the US, it's also important to have an international audience and let people know what's going on internationally. So I've done a lot of traveling in Europe, a lot of traveling in China and Japan, and I need to get to the African continent and Australia as well. So I know that this may seem a little premature. So most of you are high school students and you're thinking, well, I just, need to finish high school or, get into college and figure out what I want to do. But I think it's good to have this talk where you can start thinking about, well, what do I want to do in 5 or 10 years? What is my career path going to look like? And so I'm here to tell you that maybe a PhD is for you. It's generally known that the United States needs additional US citizens and permanent residents to get PhDs in STEM. And the end goal can be an academic position. So a researcher or a scientist at a university, a college or university. But there are many other careers that are perfect for students that pursue a PhD. And so here I just list my students. So all of my PhD students, where have they gone? And they sort of go into 3 general categories. One is the academic positions, so that's around 45%. And I list the various places that they've gone. And then you can also be a scientist in industry. So this is typically you're doing similar things to what you did in your PhD. You're just not doing it at an academic, at a university. So I list, I have a former student at Facebook, Meta. I have a student at Schrodinger, which is a quantum simulation company, MIT Lincoln Laboratories, Uber, Airbnb. But also say you get a PhD and you say, well, I have more broad interests. I'm interested in finance. I'm interested in consulting, which are generally technical careers. But you also need skills in communication and presentation skills. So I've had people go to JP Morgan, Boston Consulting, Ernst & Young. And so there are there's a wide range of careers that you can pursue after a PhD, even if you don't decide that you want to go into academics or be a scientist at a company. So next thing I want to talk about is sort of, it's a good thing, but it's a major responsibility. So there's a cost of PhD studies. The cost is not borne by you. That is, STEM PhDs are free to the student. They're not free to society. They're not free to the university and to your advisor. So your advisor typically has to raise around $500,000 for you to get a PhD. And we want to do this because we want to train PhD students and we want to get research done. So the PhD is 5 to 6 years on average. We have to pay around $50,000 in tuition to the university each year, and then stipends, particularly at Yale and other universities, have gone up recently, and they're approaching $50,000, which is not great, but you're definitely earning a living wage getting a PhD. So the other thing I want to mention is just a shout out to Yale in particular. I mentioned at the beginning that I have kind of a weird affiliation there. So I list sort of every department. I think that's something that's going to become more and more important as we tackle more complex problems. So I work on a lot of biomedical problems like cancer. There's climate problems, and these types of problems of the 21st century will require integrated and multidisciplinary approaches. And so Yale has one, the integrated graduate program in physical and engineering biology. So keep keep Yale on your radar screen as you think about graduate programs. So now I'm going to take about 15 minutes to just give you an introduction to an interesting problem that my computational laboratory is working on. And the main goal, sort of why I signed up for this, is I do want, additional students next year. So if you're a junior or a sophomore and you want to work at Yale over the summer, please apply and you can work on some of the problems that I'm talking about. So breast cancer invasion has been, or breast cancer, trying to solve it has been in the domain of genetics for a long time. And we've made a lot of progress, being able to predict sort of who will get breast cancer. But there's a lot of work that needs to be done sort of on the physics and mechanics side that needs more work. And that's sort of where I approach it. I approach breast cancer invasion from the mechanics and physics side. And so I'll just give you sort of a 2 or 3 minute overview of what happens. So this is a milk duct that's coming out of the page. So it's a narrow tube, and the lining of the tube are the yellowish, pale yellowish epithelial cells. And most cancers start as epithelia. So those epithelia will undergo what's called an EMT, which is a epithelial to mesenchymal transition, where the cells become much more mobile and they don't adhere to each other. So in the healthy state, these epithelial cells are very sticky and they sort of create a surface so that the milk duct is lined and you're not having fluids go in or go out. But this EMT happens, the epithelial cells become cancerous, they turn red in this picture, and then they can migrate into the surrounding tissue of the milk duct, which is mostly what's called adipose tissue, which is made up of adipocytes, fat cells. And the problem with that sort of breaking through this lining is that the tumor cells can then get adjacent to blood vessels and get inside the blood vessels and then migrate to distant points. And so cancer would not be a big problem. There would not be as bad prognoses if the cancers sort of stayed in the primary site. But when metastasis happens, that's when most of the poor outcomes happen. So we need to figure out if we could prevent this sort of invasion into the surrounding adipose tissue, we would have much better outcomes. And so one of the ways that breast cancer is studied sort of academically is in what are called mouse models. So one of the most common in animal models, and one of the most common animal models is mouse models. So what you can do is you can induce breast cancer in mice, and then you can sacrifice the mice at different time points and try to understand how has the invasion progressed over different time points. And so once the mouse is sacrificed, you can slice the tumor into very thin slices, stain the cancer cells, and then look at these histological slices under a microscope. And so in this microscope slide on the left are the cancer cells. The nuclei are stained this pinkish color, and then they're invading into healthy tissue on the right, which are these white larger cells, which are very densely packed adipocytes. And you can see that as the cancer cells invade, they sort of fleck off the adipocytes. So one thing that's been shown over the past 5 years is that in concert with sort of a obesity epidemic in the US, there's been much worse outcomes in breast cancer. So they've noticed that high-fat diets in mouse models, but also in human cancer patients, they have much greater invasion, much larger tumor sizes under high-fat diets. And so the general question is why? Why what's the physical, mechanical reason for the correlation between high-fat diets, larger tumors, and worse outcomes? So one kind of confusing thing is that there's so the diet is sort of on the horizontal axis. So as you have a more high-fat diet, the size of your adipocytes will grow because they're storing more fat. And on the vertical axis is the elastic modulus of the adipocytes. And so what this shows is that the high-fat diet, if you have a high-fat diet, your adipocytes are going to be stiffer. The problem with this is it's kind of paradoxical, because if the adipocytes are providing a more stiff barrier for invasion, why does the tumor grow and why is there more invasion? So one possible answer to this question is that in addition to the adipocytes becoming stiffer, there is enhanced lipolysis. So lipolysis just is a fancy word for saying that holes form in the adipocytes and they sort of pop. And so the fat pops, they leak out fat. And so what you're seeing on this histological slice, the tumor cancer cells on the left, the adipose tissue on the right, and you can see the arrows pointing to adipocytes that are smaller in size, which is indicating that they're leaking lipid. So it's sort of a sort of interesting but sort of obnoxious ability of the tumor cells to be able to pop the adipocytes so that they can invade, they can get sort of in between the adipocytes more easily, but also they can metabolize the lipid and become more active, more mobile, and more aggressive. So in the last couple minutes, say 5 or 6 minutes, I want to talk to you about where I come in. So that was sort of the background of breast cancer. I want to sort of study the mechanics of how cancer cells invade into adipose tissue. But so why would I want to do that? The reason why is because basically this statement, it's difficult to characterize the 3D sort of X, Y, and Z spatiotemporal evolution of breast cancer, because the current sort of state of the art, or not state of the art, but sort of day-to-day analysis of breast cancer invasion in mouse models is to basically just sacrifice the mice and then slice them up into 2D slices. And it's very difficult to reconstruct the 3D spatiotemporal evolution of the cancer cells through the tissue, because you have to do it. So if you sacrifice a mice at this time point, you don't have it at the later time point. So you have this mouse and this tumor at this time point, and then at the next time point, it's a different mouse. So you're not tracking the same tumor, the same cancer cells. So what would be nice is to have a faithful model of cancer invasion in a computer. And so here I'm just showing you sort of the 0th order model, which is just a 2-dimensional model where we have cancer cells on the right side, adipose tissue on the left side, and we sort of make the cancer cells on the right active, and then they try to invade into the interstices between the adipocytes. In this case, they didn't invade that far. But that was sort of the 0th order model. We now need a 3-dimensional model. And here's a 3-dimensional model. We model the adipocytes as these 3-dimensional deformable particles. One thing you'll notice that I won't go into, but you could ask, maybe in the chat, is they're sort of bumpy. Why is that? And then the cancer cells are the red cells, and then we track the 3D evolution of the cancer cells as they're migrating through the adipose tissue. So here's just sort of analogies to the 2D slices. So in the upper right is a snapshot of the 3D simulation, and then we can take 2D slices. There's a slice indicated in E, and that would correspond to the 2D slices, the 2D histological slices. So what we're interested in is just a little bit of the math and science behind it. Is in panel C and D, we want to come up with a quantitative measure for the invasion. So do you just try, if I'm going to talk about invasion, what do I mean? Do I mean do I just track the cell that's the furthest to the left? Do I track the mean, sort of the average horizontal distance that it's gone? And so we think a comprehensive way to track invasion is to find out the shared surface area of cancer cells and adipocytes. So basically what we're going to do is and I'll show it on the next slide, but whenever a cancer cell, a red circle, is touching an adipocyte, we'll basically track the surface area of the shared surface area of that cancer cell and the adipocyte. So that's shown in this slide, where I'm tracking sort of the invasion front as a function of time in panels A, B, and C. And in panel A, the shared surface area, the front between the red cancer cells and the blue adipocytes, is the smallest that it could be, because it's basically just mostly a flat wall and the cancer cells haven't invaded. But then in B, the shared surface area has grown, and because a lot of the cancer cells are now surrounding the adipocytes. And C, it's even further. So we can basically track the shared surface area as a function of time to measure the invasion. The shared surface area is depicted in panels D, E, and F. That's the surface area of the adipocytes that's being touched by cancer cells. I'm just not showing the cancer cells so you can see the surface area of the adipocytes that are touched. And the way we quantify it is through this plot. The vertical axis is the shared surface area, and then on the horizontal axis are parameters that characterize the aggressiveness and environment that the cancer cells are the environment that the cancer cells are in. And so basically invasion, no invasion is 0 shared surface area, complete invasion is 1. And so you can basically track the degree of invasion as a function of properties of the environment. So I want to leave you with 2 future directions that are going on in my lab now, so that maybe you could study them next summer. So one is that I showed you a lot of simulations, computer simulations, but how do we directly compare to experiments? I just mentioned that the sort of the mouse models, all we do is slice up the tumors, and then it's very difficult to reconstruct the 3D spatiotemporal behavior of the cancer cells. So what the experimentalists are doing, so we have a collaborator at Cornell, Claudia Fischbach, and her group, she's a biomedical engineer. They can basically create in vitro cancer invasion models. So they can have these polyacrylamide, which is a type of plastic beads, which are about the same size as adipocytes. And here I'm showing you a confocal microscopy stack of those beads. And so we just took a movie through the packing of these plastic discs. And so this is supposed to mimic a adipose tissue where you're flying through it through the individual adipocytes. So the next thing that's very important, I didn't mention it up until now, but maybe I should have, is that the adipose tissue is made up of adipocytes, so those are the fat cells, but in between the adipocytes, there's what's called the extracellular matrix. And that's a matrix of polymers or fibers that sort of give the tissue its rigidity and sort of texture. And the cells actually, the cancer cells actually crawl through this fibrous material. So them, and depending on whether the collagen fibers or the extracellular matrix is aligned or not aligned, it gives the cancer cells more or less mobility. And so we can also image using confocal microscopy the collagen fibers. And so you can see that they're a little wispy. You can see that they're fibrous. And so I'm not showing you the polyacrylamide beads. I'm just showing you the collagen fibers. And so we can image process this stack. We can image process the beads, and then we can reconstruct the positions of both the beads and the collagen fibers. And then the final thing is that we can put cancer cells in this matrix and watch them move through this matrix of fibers and beads, and have basically a biomimetic system that mimics the motion of cancer cells through real adipose tissue. So this is just puts together, after the image processing, you can create these maps of the cells moving through the 3D matrix, and you can basically capture the depth of the cells as a function of time. And then the one final thing is that I mentioned lipolysis, and this is a great future directions that in this computational model, when the cancer cells touch the adipocytes, they cause the adipocytes to leak lipid, and that gives rise to more pronounced invasion in this computational model. So with that, I'm basically at time, and I can pause here for questions. If you want to contact me later, feel free to use my email, cory.ohern@yale.edu.
Thank you so much, Dr. O'Hearn. I know I've definitely learned a lot and you've inspired our participants. So like you said, we're going to get into some of the questions asked by the chat. The first one is, how do you stay motivated during challenging phases of your PhD program or when you're obtaining your PhD?
That's a great question. So there will be, I'll just echo the question is that there will be challenges. So it's a 5 to 6 year process. I think one thing that I was always good at is looking to the future. I was good at being motivated now of a future outcome. So a PhD is very rewarding, and your career outcomes are so great after the PhD that you can sort of postpone enjoyment now, really hunker down, do a lot of research and studying, and the future outcomes will be great. But I will also say that during the process, it is all fun and games during the process. It's just sometimes it can be a little bit tedious if your experiment's not working.
Thanks so much. So another question we had asked was, what skills gained during your PhD do you find the most valuable in your current role?
Yeah, so one thing about, so I would say an undergraduate degree, and this is not always true, but an undergraduate degree sort of scratches the surface. Like what's good about undergraduate is you can sort of figure out what direction you want to go to. But like, even if you join a company after your undergraduate degree, you still need to be trained for that company. So the PhD, if you're going to go and continue a PhD, it's really where you become an expert in a particular skill set and a particular discipline. So for me, that was computational studies. So I became expert in computer programming, expert in mathematics, data analysis, statistics, and that was all learned. And you have plenty of time to learn it because you're there for 5 to 6 years, and then you become an expert, and then you can go on and succeed in your future career.
Okay. Another question we were asked is, what platforms do you use to model cells and how has it changed over the last 20 years?
Oh, so that's a great thing that I want to touch on, is that there are different types of people, so different types of scientists. So there can be computational scientists who use existing codes. So there can be scientists who just develop codes but then don't apply them. I'm sort of in the middle. So so I don't use sort of an established platform. All of my students develop codes and software that they then use to apply to to study cells and tissues. So what we use are some type of programming language, typically for very efficient coding, that would be C++, but we can also use MATLAB and Python. And you develop those codes and then you run them on various types of high performance computing facilities. So these could be just large clusters of computer nodes, or it could also be GPU, where you enhance the code so that you can run on graphical processor units.
Building off of that question, a student asked, how has the behavior of cancer cell membranes been studied in comparison to healthy adipose cell membranes?
So I would say this is the forefront of the research. So we haven't been able to sort of understand direct interactions between a cancer cell and an adipocyte. So like one thing that's very important is the cancer cell is probably sending out, chemical signals to identify an adipocyte. Then, then the cancer cell interacts with the adipocyte and then causes it to undergo lipolysis. And this sort of signaling mechanism and physical biochemical interactions are currently unknown. One thing that I will say is that there is, I guess one thing I will say that's very interesting is there is something called like good fat and bad fat. And if someone becomes fat sort of too rapidly, that's when the fat is more prone to the cancer invasion. So sort of more prone to undergo lipolysis, for example, once the cancer cells interact with these sort of fat cells that have been increased in size too rapidly.
Okay, the chats are coming in rapidly. We have a question that says, what or how does one make computational models of diseases through a system like the way you did for breast cancer, and why is it practical for finding cures?
Yeah, so the one emphasis that I was saying in this particular presentation was currently we cannot visualize. So ideally what we would want to be is we would want to have a little tiny camera. We want to put a camera in a mouse, for example, or in a human, but let's say a mouse. And we want to watch the interactions between cancer cells. We want to watch the cancer cells invade the adipose tissue. This is not currently possible. Currently, we only have access to basically two-dimensional slices of a tumor on a mouse that's been sacrificed. So one possibility is, well, maybe if we develop a computer simulation, then that could be your camera, because potentially a computer simulation can recapitulate the process that would happen when a cancer cell is invading adipose tissue. The caveat is, well, if your computer simulation is not based on anything, if you just made it up, then it's just like a cartoon. Cartoons aren't real. Maybe they seem sort of real, I don't know. So we have to come up with ways to validate the computer simulations. And that's what I described in the sort of the future directions, where we're first going to validate them to these in vitro experiments, where we build sort of an artificial tumor, where we have these plastic beads. They serve as the adipocytes. We put in the collagen matrix, which serves as the extracellular matrix in the adipose tissue, and then we put cancer cells in. And then what we can do is we can image that entire process as a function of time, and then the computer simulations should exactly replicate this biomimetic system. And if that is true, we've solved the problem. We now have a camera which can track the 3D space-time trajectories of cancer cells in real environments.
Speaking of solving the problem, a student asked, how far is our ability to effectively cure cancer?
Yeah, so I'm the problem with that. That was in my title. So we can't cure cancer, but how would we expect to cure cancer if we can? So this is I'll just throw the question back. If we can't faithfully simulate cancer, then how can we cure it? Like, if we don't know what happens, if we don't know how cancers invade tissue, if we don't know how long that takes, if we can't replicate that process as a physical scientist, I would say, how can we possibly be able to cure it? And so that's why I'm motivated to do what I do, because if I can replicate it in a computer, I think I can start to develop cures for it.
And building off of that, a student asks, how are researchers able to verify findings from computational approaches?
Yeah, that sort of builds on the previous question, is that the in vitro experiments will have basically the depth of each cell as a function of time. And so you can, for example, plot a probability distribution. What's the probability that a cancer cell has invaded to a depth d as a function of time? And so that's a probability distribution. And so if the computer simulations can exactly replicate that probability distribution, then you have a faithful simulation and you've recreated the in vitro experimental results.
Okay. We had a student that had a question about if you had any guidance on how to balance research, coursework, and other responsibilities during a PhD program.
Yeah, so that's a good question. I want to emphasize here, again, don't get stressed about doing PhD coursework yet, because that's another, maybe 6 years away. So I want to just emphasize that undergrad is different from PhD. So undergrad, you focus on courses. You typically need anywhere from 30 to 40 courses to graduate in an undergraduate program. And so you focus on courses and doing homework assignments and projects. And in your PhD, it's going to switch. So you'll do a few courses in the first year, but you're not doing courses the whole time. You're just doing courses at the beginning to make sure you have sort of, capstone knowledge to proceed. But mostly what you're doing in your PhD is you're doing independent research on your own. You can ask a friend, you can ask your advisor, how do I do this? That's fine. The interesting thing is they're not going to know the answer, because you're doing independent research that no one knows the answer to. And so your job is to do that. And so I would say the balance sort of works out, that you're not focusing on courses, you're typically focusing on research, and you have this constellation of people around you to help you, and to, your advisor, your fellow students, postdocs, to help you carry out this independent research.
Okay, so I think we have time for one more question. Is there a specific direction you see cancer research moving toward in the next decade? And for students who want to get involved with this, how would they be able to?
Yeah, so I think it's going to be a common. So I sort of disrespected genetics. I don't mean to do that. I'm just to make a point, I disrespect it. So far, basically, all we really know about cancer is genetics. If you have this gene, you're more prone to cancer. I want a more physical, mechanistic explanation of cancer. How long does it take the cancer cells to invade? What's the size of the tumor? Asking questions like physicists would. What's the pressure? Like, what is the pressure of the tumor? And I think that can go hand in hand with genetic approaches. And so there's all sorts of programs, funding of like the physics or engineering of cancer. And I think that's a really, so like, who would have thought 10 years ago that physicists would be studying cancer? Like, what do they have to say about cancer? And so I think that's a really exciting direction in the next, say, 5 years, 10 years.
Okay. Thank you so much. That concludes our time for today. I want to thank Dr. O'Hern for his amazing presentation, and we appreciate everyone here for attending our webinar for the 2024 AEOP series. Our team at AEOP is always excited to hear and share your stories. If you want a chance to win some free AEOP merch, submit photos of yourself in our AEOP gear with your AEOP mentor to the email in the chat. You can also DM us with your photo on any of our social media platforms. Our username is AEOPIANDF. We'll also put that in the chat. Thank you again all for joining us today. Our next webinar is July 23rd from 3 to 4, featuring Dr. Artie Guttom from Walter Reed Army Institute of Research, who will be presenting integrative biology approaches to diseases and military relevance. We hope to see you all there. Thank you.